Okay. Right. So what is bioinformatics? Bioinformatics is very clearly a combination of two words, biology and informatics. So if I were to take like a proper definition, which was given by the National Institute of Health, so that's something like the ICMR, or Indian Council for Medical Research in India. So National Institute of Health is the same counterpart in the US. It has taken the four, it's the forerunner for a lot of these definitions and things like that. So a lot of times you'll be seeing me quote what they said. So what they essentially say bioinformatics is research, development and application of computational tools and approaches for expanding the use of biological, medical, behavioral and health data. Okay, this would include the ability to acquire. So you have to acquire this information. You have to store this information efficiently. You have to organize it, right? Just by storing it in a, so organizing it would mean that if you have lots of clothes, you can store the clothes by shoving them inside a almirah, right? Or you can organize them in racks, your shirt separately, your pants separately, right? Then you have to archive. Some of the clothes that you would be wearing in summers are of no use right now, so you'll archive them at the back of your almirah, right? So something same happens with data as well. Something same happens with articles as well. The old articles are archived. Old data which is outdated is often archived, and the newer data is more readily accessible. Then once you have done all these things, you can analyze what kind of data that you have. So for example, if I were to continue with the analogy of your clothes, you can analyze, okay, I seem to have a lot of red color t-shirts, and I would like to have changed the kind of t-shirts that I want to wear. So you can make an analysis like that. It's just an example. Similarly, you can analyze the kind of data which is out there. So for example, let's just analyze some of the data which is being put out there. So in terms of bioinformatics, is there something currently that we all are suffering from the past 2 years around which you can expect a lot of data to be out there? This is a question to the
Sir,
class, please.
COVID-19.
Right? So COVID-19, what kind of data that people are looking at COVID-19? So let's see, I'm just searching for COVID-19. So when I say COVID-19 in Google, right? So this is a database. You're going to have all these kinds of stories, right? So one data that always comes and is very pertinent is this data of number of cases, right? Can you see my screen?
Yes, sir, we can see it.
Yeah.
Yes,
Right?
sir.
So this is one kind of data, how many people are getting infected in what locations, right? So this is data which is being live put out there, and it's updated continuously. But when we talk about sort of more biological information, COVID-19 has different kinds of strains, and what you can look for is COVID-19 sequence database. Okay, and there are a couple of sequence databases. The first one, as you could expect, comes from NCBI. This is again a National Center for Biological Information from NIH, National Institute of Health, and this is how it essentially looks like. Again, if you can see, that's where this data is hosted. So this is an NCBI SARS-CoV-2 resource. So what all does it have? This has about 3 million nucleotide records. What does this mean? This means that 3 million sequences of SARS-CoV-2 virus have been sequenced, right? And all of these would be lying in different kinds of lineages, alpha, beta, delta, omicron, right? Things like that. There are about 200,000 or 2 lakh different articles that have been published around this in PubMed. And similarly, about 2.5 lakh articles have been put in PubMed Central. So these are basically the same versions, like two different versions of
Sir,
PubMed. Yes.
these are 3 million nucleotides or 3 million genomes. Like
These are
if there have been 3 million different viruses which have been sequenced, then
not 3
that's
million
why
different
I'm
viruses. So
asking.
these are 3 million samples. So I can click on it. Okay. I'm going into it. So it's kind of slow, but these are 3 million different kinds of samples, which might be the same virus again and again. Okay. So I
Okay, sir.
encourage you
Okay.
to just explore this website by yourself. And if you have any questions, I can talk about it. So similarly, another one, which is slightly more updated, which is away from the US, but I think this one is based out of UK. So this is called GSIAID. And here it is, you can see the sequencing. They have about 6.7 million genome sequence submissions. So these many different genomes have been submitted, right? And for example, they have some information about the Omicron virus. So this is basically, if you want to know about anything to do with COVID, this is the most updated and the most, having the maximum amount of data. And here you can see what are the different kinds of features of the database. So you have some place where you can look at the mutations of the different COVID viruses. You can have mutations for other viruses which cause flu or influenza, right? There is something called genomic epidemiology, so on and so forth. So you can see this one is a very targeted database for COVID-19, specifically in the context of this current pandemic. Now, if I were to look at not just COVID-19, COVID-19 is just one virus. Just like this, there are many other different kinds of viruses. So there are databases like this. So this database is called the VIPR. So this database essentially is for all the different kinds of viruses which are out there. So virus pathogen resource. It has it basically catalogs all the other different viruses along with SARS-CoV-2, and the kinds of information that it has can be seen here. So you can get the sequence of the different viruses, you can get what strains those sequences belong to, you can look at immune epitopes. So these are basically how these viruses are actually recognized by the human body. You can look at the 3D structure of the proteins of the virus. You can look at what kind of host data, or basically what human data is sort of what kind of human proteins might be working with the virus and making it available, or making it work in the body. Then they have data about antiviral drugs, so on and so forth, right? What we are more interested in the form of molecular biologists and bioinformaticians is the data which is specifically to do with the information, the biological information that you can get. So this kind of biological information that you can get is through the sequence of the virus particularly, and you can do these different kinds of analysis on this data, and which is what, by the end of this course, I hope that when you look at a website like this, you will be able to actually understand what each one of these sequence alignment, phylogenetic tree, sequence variation, blast, all these things mean. And then you can use basically these databases more efficiently. So going back to the lecture, um, what is bioinformatics? It is basically the ability to take a lot of data, organize it in a database, and allow you to analyze that information effectively in order to make some kinds of conclusions, right? So very often bioinformatics is sort of put juxtaposed or put compared with what's called computational biology, and I would say that they are two faces of the same coin. Computational biology being more development of applications at the end point of bioinformatics. Essentially, once you have organized the data into databases and you can pull that data from these databases, computational biology allows you to work on that data to develop applications which are essentially analytical in approach, which essentially apply some kinds of mathematical modeling, computer simulation to understand, to basically make a model out of how biological information is interacting with each other. Okay. So in this course, particularly, we are going to learn about storage, organization, and algorithms to analyze biological data. So when we think about
Sir,
bioinformatics, I have three
sir,
questions that I would
computation.
like you to think about. Yes, Ratnesh.
Sir, I'm saying that like computational biology is somewhat related to systems biology. Is it that like?
Systems biology is something completely different. Ratnesh, I can answer that question after the class. Okay.
Yes, sir.
Yeah. So bioinformatics, when you think about bioinformatics, these are the three questions that come to my mind that you should be thinking. Why do you think there is a need to store information? This is quite straightforward, right? Anybody wants to try this question?
Maybe because there's a lot of data that has come up as the improvement of our knowledge about biology has improved.
So because there's a lot of data, so you need to store that data. Okay, that's good. So what kind of information do you think we are storing in biology? What kind of data are we particularly looking at?
It could be like the structures of various molecules, their conformations, or what the properties or the functions they do.
Okay, what else? So that's one kind of
Genome
information.
sequences,
Yes. The genome sequence of what?
any biomolecules like nucleotides, RNA or DNA.
Okay. Besides
We
RNA, DNA, do
Also,
you have other kinds of biological molecules
the
as well?
protein. We can store
Proteins,
the
yes.
sequences of proteins, compare the different sequence, like like for example, lactase protein in humans and in I guess bovines is slightly different. So
Yes,
we can
good.
compare that
What
and then
else?
look at the
What
folding
else?
of this
Yeah, yeah,
protein
yeah.
and check
So you
efficiency.
can
Yeah.
look at DNA sequence, RNA sequence, protein sequence. You can look at
Yeah.
folding
Compare
information. You
differences,
can
yeah.
look at what kind of function that is associated with these molecules. What else?
So
We can also find
databases of metabolites
repeats.
and metabolic pathways.
You can look at metabolites and metabolic pathways because at the end of the day, everything is actually metabolites, right? Small molecules. And when
Yes,
we
yes.
talk about DNA, RNA, protein, these are nothing but polymers made of small molecules, right? What else?
also
We
I
can
guess
look at
we also
Functions of genes.
Okay, functions of genes. I guess it would be nice if you could raise your hands and then I can sort of, easy to answer in class. So Justine, probably you can say.
yes, sir. So I guess we also store information about medicine and what all their functions do, because I guess for COVID-19, we ran various supercomputer simulations to make various permutations of different medicines to see which one acts for COVID.
Okay. Okay. So you basically are now talking about interactions between molecules. So you are saying, okay, there are small molecules which are drugs, how these drugs could be interacting with proteins of a virus. Very good. Please keep your hand down. Dave?
Yes, sir. So we can look at for example an RNA or DNA sequence and look at repeats, like
Okay.
specific 3, 4 more repeats, and find certain specific regions of the DNA or RNA in different organisms.
But would you want to store that information or that information is inherently there in the sequence that is stored? This is
We can
some
also
analysis that you are doing,
store
right?
that information. Yeah, we can store that information under some analysis section or something like that.
Okay. Okay. Ratnesh? Dave, please keep
Sir,
your
we
hand
can
down.
store we can store at a particular time genes or any a specific part of a genome of an organism is at a specific location in the space inside the cell, so
Good,
we
good.
can
Very good. Very good.
store the
Good.
spatial
Got
location
understood. Understood. Good,
of the
good, good, good.
genes.
So you can also uh, not only look at the structure of proteins, but you can also look at the structure of the genome itself, the DNA itself. Very good. So I think Dhwani, quickly, I have two more
So we
that you can
can
answer.
draw phylogenetic trees also by having different genetic strains and even figure out evolutionary history.
You
So
can figure out evolutionary history because in biology, the only theory in biology that exists is actually evolution, right? Deepak, what about you? Vishantam, after Vishantam, I'll have to stop this, right? Because I have to go move ahead with the lecture as well. Yeah, Deepak, please go ahead.
Sir, I guess we store the data about how the viruses are spreading. So whenever we encounter a new one, so we have a proper strategies to encounter that.
So how they are
Means
spreading, what would it be? So this would
worldwide,
be,
worldwide. How the what are the patterns in which uh, a
so this
particular
would be
virus is spreading?
good. So this is called epidemiology. This is basically how a disease is spreading, right? Okay.
Yes.
And last one, Vishantam. Deepak, please keep your hand down.
So like we can even have like databases of various phenotypes and say like symptoms of medical diseases so that
Okay.
like people
So
can have
basically,
something
when
to.
you can have a mapping of exactly what all aberrations inside a molecular system leads to phenotypic aberrations at the level of the organism. Very good. So,
Yes, sir.
is there something special about all this information that we have talked about? Is biological information different from, let's say, physical information or chemical information or mathematical information?
It
Think
keeps
about it.
on changing,
So
maybe
I'd
improving.
say it can't be derived.
It can't be derived. Okay.
It keeps on
Sir,
changing.
biological information like huge
It keeps on
databases,
changing.
like it doesn't follow the same rule always. Like in physical world, there may be some formula to get all
Same thing.
the
You're saying the same thing
uh,
Vishantam was saying,
yeah.
that you can have
And so
generate.
one more thing, sir, it evolves. So physical information and chemical information doesn't evolve, but biological information can evolve with time.
Exactly. So these are the different kinds of information that you already talked about. But this is what I actually was going towards, and I think you hit the nail on the head. When we actually talk about physical sciences, right? So we can go from subatomic strings, like string theory like things, all the way to celestial bodies like planets and black holes, right? But you have to really understand biology does not happen at the level of string theory or electrons, protons, neutrons, or biology is also not happening at the celestial level. Biology is happening at a very restricted physical scale, whereby it goes from biochemistry, that is how different kinds of biomolecules, chemical biomolecules are interacting with each other to form large structures and form large molecular machines, which come together to form a cell, and all these cells can come together to make a big organism, and a lot of these organisms can actually evolve together and form populations that are interacting with the environment, right? So biology, unlike physics, mathematics, and chemistry, is only happening at just a specific cross section of physical sciences. And unlike mathematics, where we sort of invented a language, in chemistry, there is a language of molecules themselves, right? Atoms and how they interact with each other. In case of physics, it's the mathematical equations that we can model the universe around. Biology, we are very fortunate because in the case of biology, there is already a language, which is the language of nucleotides. So everything that is there in the biology, it is sort of that works in biology, is encoded in the form of nucleic acids, right? Because the first step when we talk about biological information is when we talk about the biological information is the fact that you have your information organized in the form of DNA, which basically copies itself and passes on from one generation to the other generation. This DNA copy basically contains programs or genes which are transcribed in the form of RNA, which are translated in the form of protein that interact with different kinds of metabolites, right? So this is the basic information, and all of this actually information has been evolving. The only theory in biology is evolution, right? In physics, you have theory of motion. Yeah, you have theory of Newton's laws of motion. You have the theory of gravitation, so on and so forth. In biology, there is only one theory, that is theory of evolution. You had some last common ancestor which has a set of genes that could allow it to replicate, right? It could replicate, right, from one cell to two cells, and so on and so forth. Within this, there were lots of programs responsible, and this is how the information flow of information happened. That they were proteins that were basically interacting with metabolites that were available in the surroundings, and which was basically leading to the entire process to self-propagate. Okay, so you need proteins to perform functions such as transcription, translation themselves, and replication. And these things can be organized in different kinds of cells. The older kind of cells or prokaryotic cells basically do not have a nucleus. So this entire set of information flow is happening everywhere around in the cell. Then as the bacteria and these non-nucleus organisms evolved, they became more complicated. Then they wanted to have a separation of information storage, that is your nucleus, and where the actual most part of the cell's work happens, which is the cytoplasm. And that's how you got the eukaryotic cells evolved, right? And as the eukaryotic cells became even more complicated, you had the process of copying of genes into RNA getting more complicated. So you started to have what's called a pre-RNA. You had something called splicing, which enhanced the number of different kinds of proteins you can be making from the same gene. And there was some regulation in the context of when and how these RNA molecules are going to be exported outside the nucleus. And once you even have the protein, that protein itself can be decorated with lots of different kinds of chemical modifications. These are called post-translational modifications. And once their proteins have been decorated in different forms, different proteins can also come together to form more complex molecular machines. And these molecular machines, at the end of the day, either interact with each other or they work with metabolites to establish how and what a cell is doing. Okay, that was a long monologue, but this is what all of this at the end of the day comes back to the fact that all these connections, how these things are working with each other, is all encoded in the language of DNA. It's encoded in the language of ATGCs of the genes. It's encoded in the language of different kinds of RNA molecules that are made from the genes. Then how those different RNA molecules are getting translated into the form of proteins in the form of 20 amino acids, right? And how these 20 amino acids can then get different kinds of decorated modifications, and how these protein structures in 3 dimensions are actually, like working like molecular machines, which can then interact with other proteins or other metabolites. All right. So we are very fortunate that
But
at
sir,
the end, all
sir,
these
when
rules are encoded in the form of ATGC. Yes, Ratnesh. So one thing
sir,
that
I'm
we
saying
have with,
like when we
go ahead.
talk about epigenetics, so then is it just this a sequence of nucleotides sufficient for including all the data of biological systems?
It is not sufficient, but what I want to and what I wanted to motivate through this entire thing is that if you most rules in biology are encoded in the form of these letters ATGC. So you have either ATGC, this is the kind of information that you're storing, or it's the information about modifications on this ATGC, which is what epigenetics is. I have repeatedly told you that epigenetics is a more advanced topic, Ratnesh. If you want to pursue this questioning, we can do that outside the class. But epigenetics is also modification of nucleotides. So currently, there are these four nucleotides, ATGC, right? But you can chemically modify them to actually leave marks, which are called epigenetic marks. But they are also, at the end of the day, marks on these letters themselves. Okay, and this information can be stored. But at the end of the day, the language that you have in biology is all mapping back to these ATGCs. And the reason that I wanted to motivate this is to answer the question is that what is something special about biological information? What is special about biological information is that all of it actually goes back and can be mapped to this language of ATGCs. And that's why there is this whole a lot of these databases that we are going to work with, a lot of these bioinformatics algorithms that we're going to work with are going to essentially be dealing with information in the form of this four letter codes, ATGCs, or how these four letter codes map to amino acids, which are 20 amino acids. So there are 20 letter codes there, right? But there is a very specific syntax of the language that we have in which the biological information can be described. Is this clear to everybody?
Yes, sir.
Yes, sir.
So this was just a sort of a beginning for the molecular biology primer that I would like to very quickly go through with you guys. I hope that most of this has been covered already. If some of the parts have not been covered, I can go into details. But this is like the slides are actually quite self-explanatory. So there are very large number of slides here. So I'm just going to briefly glaze over them. This is just to ensure that all of us are on the same page and just to revise the basics of molecular biology so that we can uh, when we go and apply those principles in the form of informatics, everybody is on the same page. Okay, so I'm going to talk about a brief history of molecular biology, fabric of life, which is basically DNA, right? What is the genetic material, what genes do, what kind of molecules code for genes, then what about the structure of DNA, how is information transferred from DNA to proteins, how proteins are made, how do we analyze DNA, and then again I'm going to come to the question of why do we need bioinformatics. Okay, so let's hope I can cover most of it in this lecture. Otherwise, I'll continue this on Friday. That's tomorrow. So a brief history. First, you had people like Robert Hooke who discovered that organisms are made up of cells, right? Then people like Matthias Sheldon and Theodor Schwann, they expanded the study of different kinds of cells. So Schwann is actually the Schwann cells are some specific kind of cells that are found in our brain. So they were able to say that not only are we made up of cells, but we are made up of specific kinds of cells. Then there were experiments from Gregor Mendel, who was able to show that characteristics or traits of an organism can be passed on from one individual to the other individual, so from parents to their children. And these traits can be of two kinds. They can be either dominant traits or they can be recessive traits. So dominant traits are traits which are more likely to be passed on, and recessive traits are traits which are less likely to be passed on. Then when people started to look more inside the cells, they were trying to hunt for this information that is being passed on from parents to children. And there they discovered John Friedrich Michener actually discovered something called the nuclein, which is the DNA inside the cell. It resides in the nucleus of the cell. And there were a series of experiments that discovered the composition of nuclein, which was made up of nucleic acids, the ATGCs, which stored the information. And simultaneously, they were also able to find proteins and its monomers, which are basically amino acids. 20 of them have been were identified by then. And now I think we have about 21 or 22 different kinds of amino acids, which are known across all the proteins families. Um, then it was shown that different kinds of amino acids can be linked together to form proteins. So essentially showing that proteins are nothing but polymers of amino acids. Um, then it was shown that different kinds of genes are so basically your DNA is made up of repeating units or like adjacent units of genes, and these genes are discrete units. So it's not like that multiple parts of the DNA encodes for one information, one program that is required for performing a function. They found out that there is a specific part, a unique discrete unit where the information is stored, and that discrete unit is called a gene, which is passed on. Once they discovered the discrete unit in the form of a gene, then people started to discover also the copy of that gene, which is the RNA molecule, right? And then there were a lot of experiments where they were able to show that genes actually make proteins, so discrete units make proteins. Uh, then at the organismal level, they were able to show that actually the information that is there in your DNA, it is having certain kinds of patterns. And this is, I would say, this is one of the first bioinformatic approaches of looking at molecular biology data. So was this researcher called Edwin Chargaff, who basically saw that the composition of the individual monomers of DNA have a certain pattern. And because of this pattern, they were researchers such as Watson and Crick. They were able to suggest that DNA is actually double-stranded because the amount of adenine, that is A, was always equal to the amount of thymine, which is T, in the DNA that was extracted. Similarly, the amount of guanine was equal to the amount of cytosine, right, which is base pairing with G, right? So GC makes a bond and AT makes a bond, and that's how you have DNA. Two linear strands of DNA can be uh, complementary to each other and can physically bind to each other and form what's called this double helix. So this is one of the most famous pictures in molecular biology. So when I say equal numbers, I'm actually talking about the molarity, the moles. So the moles of A that are going to be there, adenine that are going to be there inside the nucleus of a cell is going to be equal to the moles of T inside the cell, and moles of G is going to be equal to the moles of C, and together all the moles of ATGC together form the entire uh, can give you the entire molecular weight of a DNA molecule that has been extracted, which basically goes to show that ATGC is is the composition of a DNA molecule. Okay. So then after this, after the structure of DNA was understood, after the complementarity was understood, people started to make a hypothesis, and they were able to show that actually RNA molecules are nothing but copies of DNA. They were able to show that these RNA molecules can actually go and bind to large macromolecular machines called ribosomes, where they produce proteins. All right. Different kinds of techniques were found during this time, whereby people were earlier, so before like about say 1950s, and during this time mostly people were looking at biology from the lens of chemistry. So when they say that they were able to figure out the composition of DNA, that was essentially doing a chemical experiment, something that you were doing in class 11th and 12th, where you would take a salt of strontium, right, or salt of calcium and put it in a flame. And according to the flame color change, you could say, okay, what kind of salt it was. So something similar was the kind of instruments that people were having earlier than 1950s. So they were only able to talk about chemical compositions and not particularly exactly how they're organized. But as after 1950s, more technologies came about, and they were able to now go into the more deeper investigations. And one of the most interesting experiments that led to the sort of pioneered the field of molecular biology was the ability to isolate DNA. Okay, so you can isolate DNA uh, actually in a test tube. Then after isolating DNA, you can cut the DNA using enzymes called restriction enzymes, which are essentially enzymes used by bacteria against viruses to cut up the viruses. So that's like the bacteria's immune system. So you can extract those enzymes and you can cut the DNA into small pieces. And once you have cut the DNA into small pieces, you can run it. You can physically, because the DNA is of a certain charge. So can somebody tell me what is the charge of a DNA molecule?
Negative, sir.
Negative, sir.
Why is it negative charge?
Because the
Uh,
because of the
because of the phosphorus molecules, phosphorus atoms there, like, sir, I don't remember exactly, but
Okay, anybody remembers?
because of PO,
Because of PO what? Ratnesh, you were saying PO4 minus,
yes, sir, PO4 minus.
right? So basically the DNA, the backbone of DNA is a phosphodiester backbone, which is essentially phosphate combined with four oxygens, right? PO4 minus. And oxygens have electron donors, so there's a lot of electrons that are there in the DNA molecules available. So the DNA is predominantly negatively charged. So by using this information, what you can do is you can run a DNA on a gel where you can basically have a positive charge on one end and you have negative charge on one end. So if you were to load the DNA close to the negative charge, then the molecules, because they're inherently negative charge, they are going to be repelled, right? So if this is negatively charged, this end, and this end is positively charged, and they are going to go basically going to get attracted towards the positive charge, and these fragments are going to move along this gel. And because the shorter fragments are lesser in mass, they would move faster. The longer fragments, they have much more mass, so they are going to be slower in the gel. And you can basically resolve DNA fragments like this. So this led to the event of recombinant DNA technology. And I think most of you have gotten exposed to this, right, in the molecular biology course. Can you confirm?
Yes, sir. Yes, sir. We have.
Yes.
Okay. All right. So then after this, there were experiments whereby people were earlier looking at simpler organisms like bacteria, but then they thought that, okay, there's actually what happens in more complicated organisms which have a nucleus. So they found things like mRNA splicing, which basically allows the same set of genes to make many more different kinds of proteins, and I'm going to talk about it later on. And they were then this was around the late 1970s, and then came about a big revolution. Um, and this was basically the development of an automated sequencing machine by Leroy Hood. I happen to know him personally. He was my PhD advisor's advisor. A very nice man. So he basically pioneered the technology which allowed us to again look at what the basic language or basic letters in biology, which is ATGC. And this technique of sequencing basically allows you to look at the composition of a DNA molecule. And because RNA molecule is nothing but a copy of the DNA molecule, so this allows you to also look at the composition of RNA molecule. And because DNA molecules and proteins have a specific relationship of three sets of DNA molecules encoding a codon or a specific code for an amino acid, you can, if you know the composition of ATGC in your fragment, in your DNA, then you inherently can map it to the protein space of the 20 amino acids given your translational rules, right? So as soon as this invention happened, the invention of the automated sequencing mechanism, um, there was a explosion of information in terms of in terms of biological information in the case of molecular biology, where scientists were trying to understand what is the sequence of DNA of different kinds of organisms, and therefore, as soon as this technique was evolved, it was released. The first project that the most important project that people started to do was to understand what is the composition of DNA in our own body, in our own cells. It was started in 1990. The first version of it was finished in 2001. And actually, only last year, the human genome was officially completed. So basically, it was took 30 years to finally confirm what is the composition of all the nucleotides, ATGCs, that are found in our cells. And I would say at some level, this is still an ongoing effort. And because the DNA mutates rapidly, um, this is information that we'll still be looking at. More than that, people also started to look at what points of time different kinds of RNA molecules were being made inside the cell, which is a dynamic process. And then if you are can take, say, different kinds of RNA molecules are being made inside a human cell, then you can also start to map the differences between two different kinds of human cells. So it's remarkable that all of us at some point of time as human beings existed as a single cell inside our mother's womb, right? And then from that single cell, all the different cells in our body have been made, but they all come from the same set of DNA. So the next set of questions that people started to ask was what makes these different cells different? In order to do that, they were not only they know that DNA is the same, but then that essentially means that the set of RNA molecules and thereby the set of protein molecules which are present in the different cells would be different, and that is what they have found. So people started to generate a lot of information around this. And this is another big challenge in bioinformatics that I would be talking about. And then we would be understanding how do people do this, and what are the technologies, and what are the different kinds of techniques that people use informatically to answer these questions. Um, Naturally, going ahead, although the Human Genome Project was announced in 1990, people started to do much smaller genomes. So in 1995, the first bacterial genome was sequenced. Interestingly, the first bacterial genome sequenced was an influenza genome. So we are looking at coronavirus right now, which is also causes influenza. But there is also a bacteria which causes influenza, which is called H. influenzae. I please, I hope that everybody goes back and maybe Googles that. Maybe you can Google that right now. The what was the first bacterial genome sequenced, and can tell me by the end of the class the name of it. So when they did that, when they were able to sequence the first bacterial genome, then they automated the process of automated sequencing developed by Leroy Hood by several orders of magnitude. And they were able to come up with a robotic instrument, which is what we typically are using for big large scale sequencing projects currently, where you can not only sequence a bacterial genome. In fact, you can sequence thousands of bacterial genomes in a single day. You can even sequence like tens of hundreds of human genomes in a single day. So all these things started in 1995, and this is something that I'm going to talk about. So a large part of one whole module of the course will be dedicated on the Human Genome Project and the different kind of technologies that came out of the Human Genome Project, and how we are actually using these technologies to answer questions of bioinformatics, particularly in the context of data generation. So then after the bacterial genome, they looked at the other smaller eukaryotic genome, so yeast. So yeast genome was sequenced. So this was called the Baker's yeast genome. So this genome from this yeast is called Saccharomyces cerevisiae. This is about two orders of magnitude larger than the bacterial genome. So keep on going, generating more and more information. Um, then the E. coli genome was sequenced. There were a lot of technology developments which happened. Then they sequenced a whole organism, the eukaryotic worm called C. elegans, Cyanohabitus elegans. So it's a roundworm, right, which is found in human stomachs, which causes the roundworm disease. And then in 1999, they were able to sequence the smallest chromosome of the humans, which is chromosome number 22. So human chromosomes are numbered from 1 to 22, 1 being the largest chromosome and 22 being the smallest chromosome. So 22 was the first one. As I said, this 2001, they completed the sequence of the Human Genome Project, the Human Genome. The first version was released. In 2000, before that, 1 year before that, the sequence of uh, a model organism called the fruit fly. This is the Drosophila melanogaster. So if you leave a banana peel outside, they are like these very small flies, not housefly, but very small flies. They usually have red eyes. They're very, very small. They could sit uh, like 1 millimeter in diameter, like 1 millimeter in length. They're very small flies. So these are called fruit flies. They are very often used in biology to do experiments. So their genome was sequenced, right? So as you can see, as soon as from 1986, the sequencing machinery was developed, humans, scientists, they basically wanted to sequence more and more and more genomes. So they wanted to first sequence different kinds of organisms, right? Different kinds of bacteria, different kinds of eukaryotic fungi, right? Yeast, for example, then complete organisms like C. elegans, Drosophila, then humans, then mice, then other kinds of mammalian organisms that are typically used in the lab, right? And then they basically that has essentially given rise to all this kind of information that is being currently put in all these biological databases. Okay, so coming back to what I was saying, the fabric of life essentially is DNA, right? You have different kinds of cells. You can either have prokaryotic cells and eukaryotic cells. I've already talked about the basic difference between these two, so I'm not going to go into it. Um, a cell is essentially made up of three different biomolecules, mostly DNA, RNA, and proteins, and all of them are interacting with each other through mechanisms called together, like an umbrella term called cell signaling. I'm not going to go into details of that. We all know what a cell is. It's the fundamental unit of life. There can be two kinds of cells, eukaryotic and prokaryotic cells. A cell by weight is mostly water, 70% is water. Why is it water? Because life evolved in water, right? 7% is made up of small molecules, which is basically salts, lipids, amino acids, and nucleotides. So you can say these are monomers or these are metabolites that are existing in the cell. And about 23% or a quarter of the cell is made up of macromolecules, which are nothing but polymers of these small molecules. There are four major polymers. There are proteins, there are polysaccharides, there are lipids, and then there are nucleic acid polymers, right? DNA and RNA. There are different kinds of metabolic pathways in which these macromolecules are interacting. And something essential for the cell to pass on information is the ability to make proteins from a DNA molecule. So the first process is called transcription, where you copy the DNA molecule into an RNA molecule. And then the next process is called translation, where an mRNA molecule is copied into the DNA molecule. Okay, so this is how a cell looks like. There are certain features. I hope everybody knows these features. A cell can have different kinds of versions. Either a cell can keep on dividing, right? So when a cell is dividing, it has different kinds of phases. The first phase is where your nucleus increases in size. So this is called the S phase, where it is synthesizing essentially. And then the cells can divide after it has enough amount of DNA, when the DNA gets totally replicated, right? So this is how cells are typically. This is what's called the cell cycle. Um, so cells basically are born to eat, replicate, and then die. To summarize what this picture shows. So I hope everybody knows about the cell cycle, right? I'm just like repeating this.
Yes, sir.
Yeah.
Yes, sir.
Yes,
Okay.
sir.
Again, two types of cells, eukaryotic and prokaryotic cells. This is how they look like. Uh, there can be organized phylogenetically in three kingdoms. There can be the bacteria kingdom, there's the eukarya kingdom, and there's the archaea kingdom. The eukarya kingdom is what we belong to. It contains plants, animals, fungi, and different kinds of algae. The archaea kingdom is the ancient sort of organisms which are probably the ancestors of both eukaryotes and prokaryotes. And then you have the prokaryote kingdom, which is mostly bacteria and other kinds of single cell organisms. This is just a textbook thing, how eukaryotes and prokaryotes are different. Um, you have different kinds of cellular machinery, which so these are the major processes that are controlled inside the cell. So instead of having a brain, a cell makes decisions through complex network of chemical reactions. So that's the first part of a living organism, which is the biochemistry. So biochemical reactions. These are organized in the form of pathways because you have one reaction feeding in. So you have the substrates of a reaction on the left side, which makes products. These products are fed as substrates to another reaction, and so on and so forth. So you basically make a chain of chemical reactions, and these are called pathways because one molecular input at one end of the pathway finally gives rise to the completely different product at the end of the pathway, and these pathways are important. So these pathways synthesize new materials inside the cell. So these could be cell's own materials. So a cell essentially is going to have a membrane. It has different kinds of membranous organelles such as your nucleus, mitochondria, endoplasmic reticulum, Golgi body, membrane, plasma membrane that surrounds the cell. There is you have to break down these membrane. You have to break down these materials at all points of time. So just like a city, you have to recycle things. So a cell basically does that. And then there are signals that allow the cell to eat food from the environment. And then there are signals that allow the cell to die, basically pathways. So a cell stores all this information in the form of the genome. So human genome is about 3 billion base pairs long. Imagine every cell in your body, so there are trillions of cells, contain the same sets of genes. I'm just repeating this information. However, not all these genes are expressed. What do I mean by expressed? Not all these genes are making proteins in all the cells of our body, as I said. Different cells are making different proteins. That's why these cells look different, because proteins are the building blocks of a cell. Essentially, these are the proteins which are defining these pathways. Proteins are nothing but enzymes, for example. I mean, proteins, one of the major roles of proteins is to be enzymes. So if you have different kinds of enzymes, so different kinds of pathways will be activated. So for example, if you were to compare a blood cell, right, which is a round, small cell inside the body, not that small, and compare that with a neuron, which is a very large cell, which has a very long body. So you can imagine the pathways, which is making the membrane, right, synthesizing the membrane inside a neuron is going to be much more active as opposed to the pathway which is synthesizing a red blood cell, which is a short-lived cell, right? So red blood cell doesn't basically, it's a round cell which is made and then it dies within a certain amount of time. On the other hand, a neuron, it gets made and it stays for a very long time in your body. So these pathways are always kept active. So you can imagine the pathway which is involved in membrane synthesis is active differently in the two kinds of cells, right? So basically a cell in one form is like a factory. It collects and manufactures components. It carries out replication of different components, and then it makes a copy of itself to make another car factory, which in the form of a new offspring. Okay. So certain analogies that you can understand in the form like how the information is organized. So nucleus is basically a library of all the different chromosomes which are there uh, inside your DNA, right? Your DNA is organized in the form of chromosomes. Each chromosome is going to have a set of genes. Each gene is essentially is like a, is like a book or a recipe of making a protein, right? So this is how you can think about information inside a biological system. So almost all the cells in an organism contain the same libraries, right? The same set of books. Almost all organisms have to divide. They have to make copies of themselves. They have to eat food. almost all organisms eventually die, right? So these are certain things that are there which are conserved across all the organisms, and that's why we are basically able to say that there is an evolutionary relationship between different kinds of organism cells that are existing. Okay, so I will stop at this point of time. These are some terminologies: genome, all the genetic material of the organism; gene, discrete unit of hereditary information; a genotype is the genetic makeup of an organism; phenotype is the physical expression of a trait; and nucleic acid is a biological molecule that allows organisms to reproduce and transfer information. So yeah, so I'm going to post this particular lecture. I am sorry that I have to be slightly boring for this lecture, because again, I would like to basically hope that everybody is up to date with the molecular biology different terms, because when I will start with bioinformatics algorithms, I would assume that everybody understands what I'm talking about. So I think spending 1 or 2 lectures on revising these molecular biology terms, although it might be boring, but it would be very important and useful for the course. Okay, so I'll stop here. I'll be posting this lecture link in Moodle. So this basically I host these lectures on Google Drive. And if you have gotten the links from your previous batches, please, I encourage you to use the latest links, because I am always updating the lectures based on the feedback that I get from each batch, and also the information which is put out there in the domain out there on the internet. All right. I'll stop. I'll take a few quick questions. Otherwise, if you have the other to have to go to the other class, please, you're free to leave.